Yes, hello everyone. Welcome to my YouTube channel Basic Concepts of Microbiology. Today we will talk about bacteriophage mu. Uh, as it is, we all know what are bacteriophages. Bacteriophages means, see, phage means virus, right? Bacteria, right? Okay. So these are the viruses which feeds on bacteria, right? So these are known as bacteriophages. As we can see from here, bacteriophage, these are the viruses that invades the host bacterium cell, right? Right. One is E. coli. E. coli, right? So the phages will be known as coliphages. Right? Okay, we will see later on, right? So there are like phages for this one, blue-green algae. They are known as cyanophages, right? So we'll talk about bacteriophages. These are the viruses that invades the host bacterial cell, while muphage are those that show the properties of both viruses. We said mu for mutation, right? Here mu stands for mutation. Mu means mutation, right? Mutation. Okay. So what happens? Okay. So what are bacteriophage mu? These are the phages which have the properties of both virus as well as transposons. What are transposons? We'll see later on, right? So muphages is also called temperate transposable phages, right? Temperate or transposable phage because it causes the transposition of the genes, right, into the host cell at the time of multiplication. What does it mean? Whenever, see, viruses, they don't have their own, what do you call, machinery or replication mechanism, what do you call, replication machinery. They have only the genetic information. So what happens? They inject the genetic material into the host cell and they hijack the machinery and starts multiplying their copies, right? So what happens? These muphages, they infects the, what you call, host, and they causes transposition of the genes. That means they moves the what you call genes from one place, like they are shifting their genetic information, right, or genetic content in the, what you call, RNA or DNA of the genetic material of the host into the host cell at the time of multiplication, right? And this happens when there is a multiplication in the host, right? It can infect E. coli and may undergo both lytic and lysogenic phase. Lytic, there will be lysis, and lysogenic, they'll become the part of that host phage, and they will keep on multiplying, but they will not rupture or damage normally, right, into these hosts. But whenever they have to, under certain conditions, when they have to come out, they will follow the lytic pathway, and again go they will do lysis of the host cell, right? It is also a transposon and can cause mutation when transposes the name, when transposes, right? The name mu stands for mutation. Mutation, that is the changes in the genetic material. So what are transposable or muphage? It belongs to the family Myoviridae, and it is a double-stranded DNA virus. It attacks mainly members of species of the Enterobacteria family, and that's why also known as Enterobacterial phage mu. Their hosts are generally E. coli, Escherichia coli, Serratia marcescens, Citrobacter frundi, Erwinia, and Erwinia cartovora. All these are Enterobacteriaceae. Transposition of muphage occurs by either of these two mechanisms, either by replicative mechanism or by non-replicative mechanism, right? Okay. Fine. If we'll talk about the structure of these phases, their structure is something like this. Their head part is icosahedral, then they have this one tail, then these are tail pins. This head have genetic material, so you will have the genetic material, and this is what it is covered by sheath. It is contractile in nature. So this is icosahedral capsid, this is neck portion, and this is base plate, right? Okay, this is somewhat the structure of bacteriophage mu. So basically, if you will see, they have three basic parts, right? One is head, another one is neck, this one is tail, right? And tail, this one. Sometimes this will be also considered as tail, right? Okay, so the symmetry is of mainly, symmetry is head of the, first they have icosahedral symmetry, right? If you was like this, they have icosahedral, that is, they have specific number of vertices, right, and facets. The shape is almost a spheroid, and the diameter of this one, it is 54 nanometer. 54 nanometer, right? And their capsid is, this one. The covering, the membrane which covers this genetic material, it is known as capsid. They are made up of proteins, capsomeres, right? So these are the layers which surrounds the hexagonal head. Okay, capsid is made up of about, sorry, this is not capsid. Sorry. Capsid will be outermost part, right? Okay. Suppose this is the icosahedral head, and this will be the capsid. This will be the capsid, right? And this is the, what we call, head. So these are the layers which surrounds the hexagonal head, and capsid is made up of about 152 smaller protein subunits known as capsomeres. These are known as, right, capsomeres. Here it is capsomers. Right, these are capsomeres. Okay, fine. The neck is the middle portion of the mu phage, which acts as the joining segment of the two components, that is head and tail. Right. So it is a long, thick and contractile. It is contractile like string. It is contractile in nature because they have to inject their genetic material into the host. So that's why it is contractile in nature. Due to the presence of cross bands, size of the tail is about 183 to 16 to 20 nanometer, right? And there is a sheath. In this tail, they have a sheath. What kind of sheath? This is tail, I have told you, like spring-like structure, right? So this will compress, and the size of the tail is about this one, 183 in length, 83, and this one 16 to 20 nanometer, right? And this one 180 nanometer. The sheath of the contractile tail, which is composed of stacked rings, right? At the time of contraction, the sheath becomes shorter and thicker. Obviously, when we press the spring, it becomes shorter and thicker. So these are, this one, sheath becomes shorter, stacked rings. Then there is a tail fiber. Tail fiber, this part, right? Okay. These are the thin and thread-like structures which are attached to the large base plate. There is a base plate, and there are about 6 long tail terminal fibers. Here we can see the structure of mu-phage. This one, outermost capsid, covering of protein, which are made up of capsomeres. Then there is the double-stranded DNA, and this is icosahedral. Then there is a neck, right? And there is a tail, neck, this portion, there is a tail. Tail has a sheath which is made up of stacked rings, right? Okay, these are the base plates, and they have certain specific proteins through which they attach to the host cell surface, like N protein, right? Then there is a tail fibers and tail, and this one is a base plate. So this is the basic structure of a bacteriophage mu. Come to the genetic organization or genome organization. See the genetic material of this phage. They have all these. The outermost one, both the sides, they have this one, attachment L and R. Both are transposase binding sites. Since we have seen they have both transposition nature as well as they are viruses, right? So because of this site, they have this transposable property, right? So this site, it is left one at L and at ATTR, right? Then there are group of different genes which have different role, like regulation of transposition is controlled by the C genes, right? Transposition is done by AB genes, right? Lysis by LYA genes. Then head and tail part is framed by these ones, right? Head range again these ones, and there is a mom gene, right? And host DNA, this one host DNA, right? So they will attach in the host DNA and they will multiply, as we have seen in the very first slide, right? And they will make their multiple copies. Fine. So mu-phage consists of double-stranded DNA that is linear and non-segmented. Genome is non-permuted and consists of some unusual base pairs. That is, they have hydroxymethyl uracil, which is not normal. Otherwise, there should be uracil, and terminally redundant sequences. The guanine and cytosine content is about 35%. Sorry. Yes, guanine content is about 35%, right? So mu-phage genome encodes about 55 genes which perform, we have seen in the previous slide, different functions in its life cycle. Genome consists of 3711 base pairs. The genomic size is about 40 kilobase pair in length, with unequal lengths of host DNA at the two ends. Okay, we'll see in the next figure. And DNA segment can invert and is found in both orientation in viral DNA. Yes, this is a fast DNA present in the virus particles, what we have seen over there, right? Okay, now what is the prophage before maturation, right? Okay, this is the same what we have seen over there, right? These are the at L and at R, they will insert in the host DNA and they will start multiplying. Okay. So the function of different genes are as follows, as we have already seen over there, right? Okay. Mu-phage consists of two transposase binding site. These two transposase binding site. Right. Okay. And that binds to the host DNA and are represented as at ATTL and ATTR. These two ends are sometimes called as mu-L and mu-R. Okay. Then there is A gene, B gene, and C gene. We have seen it, and it encodes for A encodes for all the transcription events, B encodes for all the replicative transposition. C gene, it represses the expression of the transposase genes, right? While head and tail, we have seen these are responsible for the head and tail region. Right. Then there is GIN gene. It catalyzes the site-specific inversion reactions. Then there is a MOM gene we have seen over here. This one. mom gene, what does it do? It encodes as DNA modification function by converting adenine to acetamide adenine, right? Then there is a lysine, we have seen lysis or lysogenase. It encodes the lytic enzyme which causes the lysis of the host cell, right? Okay, as we have seen, they can go for lysis and lysogenic cycle, right? Okay, if we will see the life cycle of muphage, first they will attach with the host cell. Suppose it is E. coli, the host, they will attach with this N protein was there, right? So this one, the sugar, it will remember this with their receptors. They will find their receptors and they will sit over there, right? After that, they will like drilling, right? They will start injecting, and the sheath will be tail part will be contracted, right? So that, and once it is completely contract, it will shrink, and that the genetic material, just like injection, that will be delivered over here. And now they will integrate that genetic material from the this phage. They will insert inside the host genetic material, and host genetic material is responsible for like all the activities. So once they will multiply, their genetic material will be also copied, right? So this circularization, there is a series of events. There will be circularization, then there will be integration in the host cell, right? Host genetic material. Then there will be latency. There is a possibility it may go for latency, right? Okay. And after latency, they may go for, if there is a latency, then they will follow this one. Okay. Otherwise, if they are going for latency, they will go for replicative transposition. We have seen replicative, non-replicative. Then again, there will be modification of the viral DNA, like some phosphoryl groups or some other will be added, right? After that, there will be biosynthesis, their head and neck portion, everything will be synthesis, and the genetic material too. And there will be assemblage, right? Assembling, and that is DNA packaging. Finally, once it is fully matured, till now it was prophase, right? Once it is completely mature, they will go follow the lysis and they will come out. This is how they are following this what you call life cycle of muphage. These are the early genes, if they will be activated. Okay. So muphage, we have seen already these steps. Attachment is done by, the first step is attachment. It attaches to the host cell surface via lipopolysaccharide and thereby initiating infection. Then there is ejaculation of the genetic material into the host, okay, whenever that sheath will contract it. Then there will be circulation. The N protein, which is present in the virion, is ejected with and binds to the viral DNA to circularize it. The DNA ends are thus protected from the host nucleases, right? Then there is integration. There must be some early transcription giving rise to the least the Rep-C, REPC and NER repressors. These are the repressors. And to the DDE recombinase, there is a mu-A, which performs the integration. There is a flanking bacterial sequences which are cut away from the viral genome before integration. Then there is mu-phage cycle latency replication switch. So the ratio of these two genes, that is ratio of repressor C and nerve repressors, they determines if the phage enters latency or lytic cycle, right? Rep C represses early promoter, thereby establishing latency, whereas NER represses Rep C expression, thereby promoting early gene expression, leading to the onset of viral replication. That means these two decides whether the cell, they will follow the lytic or lysogenic pathway. So we have seen earlier there is a two mode of replication. One is replicative, another one is there is two mode of transposition, replicative and non-replicative. So what happens in replicative? When repressor Rep-C is inactivated, both DDE recombinase in mu-A and target DNA activator B, that is mu-B, are expressed. Mu-A is a part of transposome complex which performs recombination between the viral genome ends and the host DNA. Further, this viral host DNA structure is resolved, right? It will be cutted from that site by target primed replication, leading to two copies of the viral genome. This process is called replicative transposition, right? Selection of transposition site is performed by this gene, mu-B, right? Successive rounds of replicative transposition can lead to about hundreds of copies of the viral genome. This is how they are making their own copy, right, into the host vertical cell. Okay, mechanism of transposition in mu-phage. What happens in mu transposition? Initiates with the mu transposition initiates with the cleavage that exposes the 3' end of the element, right? 3' element will be